So for this immunological memory, so it's very important, as I said, that our body nature will have the ability to remember. Okay, for example, if like I say that if I throw you into the river, you already know how to swim, you won't forget how to swim. Are you clear? Unless you didn't practice this for 10 years, 20 years, 50 years, then suddenly I throw you into the river, maybe you can't swim. Are you clear? So the period sometimes must be long enough for you to forget how to swim in this case. Okay, so same goes to our memory B cells. Memory B cells, they are long life cells, so sometimes maybe few years. Okay, doesn't matter. Okay, but you need to revise, as I say that. Yeah, you need to do the revisions, right? I mean, okay, after 2 years, you go pick up swimming again, and then you stop for a few years. Okay, but you still have that kind of memory. So immunological memory depends on the memory B cells. Highlight the most important one is memory B cells. I will tell you guys why memory B cells and also memory T cells. So follow any immune response, memory T cells, they will group strategically in many non-lymphatic tissues, including the lung, liver, kidney, and intestine. But B cells is there. They have the gene that prevent apoptosis. Now B cells have the gene to prevent apoptosis. Basically, they don't die. Okay. So therefore, they can continue to live and produce a lower concentration antibody long after the body has overcome an infection. So the presence of circulating antibody is usually clinically to detect previous exposure to a specific pathogen. Okay. For example, if we do the HIV screening test, we measure the antibody to HIV. How you know a person actually infected by HIV? We check for the plasma antibody, because if you are not exposed to HIV, your body won't produce antibody that targets HIV. That's how we detect it. Are you clear? Okay. So if the same pathogen, say with the same antigen enter the body again, the antibody immediately targeted for the destructions. So at the same time, specific memory B cells are stimulated to divide, producing a new clone of plasma cell that produces the same antibody and also a new batch of memory B cells. So try to imagine this, guys. It's something like revision. Okay, something like revision. Okay, so first, I throw you guys in the river, first time. You struggle, you learn, you remember how to swim. Throw you in the second time. No problem, immediately you can swim. Do you need to go through the learning process again? No need. So same goes to your memory system, the memory B cells here. The memory B cells do not need to actually be activated, wait for the helper T cells, get the cytokine, get the antigen ready. It's too slow. Are you clear? So these memory B cells immediately stimulated. One is stimulated, they undergo clonal expansions again to produce a new batch of the plasma cells and also produce a new batch of memory B cells. So it means that it's something like revision. Because your memory B cells, it won't last for too long. Okay, because even though it lasts for long, it doesn't mean that all of them will survive at the same rate. If let's say I have 10 memory B cells, over a long period of time, some will die. So it means left with 2 or 1. Can you see that? But if a second exposure take place, what will happen is the remaining 1 or 2 of them, before they die, we train them to proliferate. So it means that we get replenish a new batch of the memory B cells. This is some form of revision. Can you see that? If you don't do the revision, right, in this case, after 10 years, 20 years, you don't have any memory B cell left. So you have to restart the entire process again. Can you see that? Are you clear? So once you have what we call this, the memory B cells, so the process will be very, very fast. And why we target B cells, talk about B cells, not the T cells? The most important thing here is B cells actually turn into the plasma cell secrete antibody. One of the raw antibody bind to the these antigens on the pathogen, prevent them to enter into the cells. What we want to achieve here is we do not want the pathogen to create the problem to us. Once you have symptoms, you show that you have fever, you have a nausea, vomiting, diarrhea, for example, okay, tired, fatigue. Those symptoms, because of your cell have been infected, it show you the symptom. Are you clear? So before the pathogen enter into your cells or destroy your cells, we want to block them. Memory B cell is the most important one. Are you clear? But memory T cell is important or not? It's still important because memory T cell, if let's say somehow this pathogen managed to enter into our cells, infected already the cells, then the T cells, memory T cells have to do. But again, it will give a very, very mild symptom only. Are you clear? It will give us very, very mild symptom. So why mild symptom? Because majority of the pathogen already blocked by the antibody. Just the remaining some of them manage to escape and infect the cells or body cells. Are you clear? Can? Okay. So let us look at a secondary immune response. So first exposure to the antigens is known as the primary immune response. So primary immune response takes 3 to 14 days. So that's why when you have the injections, can you see that? The second booster, I mean, injection, they say 14 days. After 14 days, you get a protection because your body needs time to gain sufficient, okay, antibody. So it's termed as a latent period. After the latent periods, then you have a logarithmic periods that's increased rapidly for several days until it reaches a peak. And then you have the decline phase. You can see that first latent, then log phase, and then decline phase. Can you see that? Are you clear? So, but a second exposure to the same antigen even after few years result in a secondary immune response. So memory B cell bearing antibody to that antigen and also memory T cell will proliferate and activate, which is much more rapid, okay, compared to primary response and have shorter latent periods and more antibody are produced. So guys, you can see that this is antigen number 1, infected first time. Take how long to reach this concentration? It takes about 1.5 weeks. Can I see that? Okay, 1.5 weeks to 2 weeks. But if you look at this concentration, to achieve this concentration for the second injection, so achieve it within half a week. Can I say a few days we managed to get this concentration, and after 1 week you can get higher concentration. Are you clear? But for antigen number 2, it's the first time you encounter it, so you can see that the same process, the same challenge actually take place. Clear? Okay, so this is a primary immune response for antigen 1. This is secondary immune response for antigen 1. The green color here is the primary immune response to the antigen number 2. Okay. So when someone is exposed to the same antigen again, so our immune system responds quickly, destroy the pathogen before they have time to multiply and cause a symptom in our body. Okay, for example, measles, chickenpox is unlikely for us to get reinfected in the future. But sometimes in some diseases, okay, for example, influenza, why someone get influenza, the next year influenza again, the third year also influenza again? So the answer here is because the variety of this virus, okay? So because they have the ability to mutate and they create more than 100 different viruses. So in this case, how we can actually inject? You cannot inject 100 times or 100, vaccine. Can you see that? You won't get infected by 100 times, 100 types, okay? So as the virus evolve, changes may occur in the surface antigen. So the slight change also may also prevent the recognitions by the memory cells. So every time is a new immunological challenge. Are you clear? So that's why sometimes we see that, how come I'm immune to Delta but not Omicron, say in terms of COVID? It may be a different strain, but they are still COVID. Can you see that? But different surface antigen. Okay. So let's look at activity 12.3. So you can see that the cell A, cell B, and cell C, and you can see this is the bone marrow. Okay, so this is the one with the C shapes. Can you see that in the blood vessel here? So the C shapes, okay, can you see that indentation? So this is a monocyte right now. This is a monocyte. So when the monocyte, okay, pass through this, okay, squeeze through the wall, the capillary, you get the macrophage. Can you see that? Okay, bone marrow also give us, this is the neutrophil. For bone marrow, you have the B lymphocyte. We have the immature lymphocyte migrate to the thymus, give us the matured T lymphocyte. So it can be cytotoxic T, it can be the helper T. Clear? If not clear, go back to my video, okay? You guys need me to write. So this is the bone marrow, the bone marrow there. So this is a monocyte, okay? The hematopoietic stem cell. Hematopoietic stem cell at the bone marrow there. Okay, so hematopoietic stem cell will give rise to the first progeny monocyte. Monocyte will develop into the macrophage. Okay, and bone marrow also gives us this is a neutrophil, right? Okay, and the maturations, you can see the nucleus take up about 80 to 90% around nucleus, a big cells in this case. So this is a B lymphocyte. Okay, and D is a thymus. So we have the lymphocyte that migrate to the thymus and then mature to form a T lymphocyte or T cells. Okay. So explain the role of the cells A, B, C in the immune response. So cell A actually is a macrophage. Okay, carry out phagocytosis, engulf the pathogen, and non-self antigens are processed, display the antigens, okay, the non-self antigen on its surface. So some of the cells which are helper T cell recognize the non-self antigen presented by APC. So the binding between the binding site of the T cell receptor and the presented non-self antigen activate the helper T cells to secrete cytokine. The secreted cytokine stimulate the macrophage to carry out phagocytosis more vigorously. Cytokine also stimulate the clonal expansion on appropriate helper T cells, cytotoxic T cells, and B cells. T cell and B cell undergo mitosis and cell division, produce their respective clones. Therefore, high number of B cell and T cell is available for primary immune response. So these activated cytotoxic T cells secrete toxic substances, for example, hydrogen peroxide and perforin, to lyse the infected cells. The remaining cytotoxic T cell may develop into these memory cytotoxic T cells. How about B cells? B cells develop into the plasma cells. Okay, some of them, so plasma cells synthesize and secrete antibody to bind to the non-self antigen. The remaining activated B cell again develop into the memory B cell. So helper T cells develop into the memory helper T cells, memory cytotoxic T cells, and memory B cell remain in the body for second exposure to the same non-self antigen. The secondary immune response is a rapid response which helps to remove the pathogen before the appearance of the symptom. Okay, so you can take a photo if you need. If you're done, just show me a next. Kenna? Next. Okay, clear? Clear? So I think that's all for, okay, this is a summary. Again, you can see that this part is a humoral immune response that involves the activation of B cells to get plasma cells and memory B cells. All antigen presenting cells now have the, to activate the helper T cells. So you can see the helper T cell activates the B cells by releasing a cytokine, activates cytotoxic T cell by releasing cytokines, and also actually activate it itself to form a more helper T cells and memory helper T. Okay, so this one you can read the lines and it's very, very simple. Okay, so and for this part,